We have a test on enzymes, and this video is going to help explain what you need to know. So this is for freshman biology, and the big thing about enzymes is that they are a type of catalyst. And what a catalyst is, it has to do with decreasing the activation energy required for a reaction to go forward. So in a chemical reaction, you start with reactants. The reactants react and eventually will produce products. But in order to produce those products, you first must reach a certain amount of energy. There's an amount of energy required to make this reaction go forward. When you take chemistry, you will learn about the bonds that need to break in this molecule and reform in order to make the products. And in order to break the bonds, energy is required. So in order for that reaction to go forward, you must reach a certain activation energy. Catalysts will lower the amount of activation energy required. So it's like they take a shortcut. So this is for all chemistry, not just biology. Catalysts make it less energy required. They take a shortcut in order for the reaction to progress. The reaction can happen without a catalyst, but it takes a lot longer. So it's easier for the catalyst with the catalyst. And catalysts in living things, specifically in living things, are called enzymes. Enzymes are biological catalysts, and they're always made of proteins. So the enzyme in general has a certain shape. We'll talk about the shape in a little bit. The shape is important. And in a certain part of the protein, enzymes are proteins. Proteins do everything. And a certain part of the enzyme, there's something called the active site. And the active site is two things. It's where the substrate is going to fit in. It's also where the reaction is going to take place. So the substrate is going to fit into the active site. And that's where the reaction will happen. And the enzyme is going to speed up the reaction because it's lowering the activation energy required. It could happen without enzymes, but it would take too long, and that would not be conducive in living things. So you wouldn't be able to survive without these enzymes speeding up the reactions. So the substrate, you can see its shape is complementary to the enzyme, so it's very specific. Each enzyme acts on only one type of substrate because the shape is specific. They fit together, you could say, like a lock and key. When the enzyme and the substrate bind together, you can consider this whole thing called the enzyme substrate complex. So this is called the enzyme substrate complex. This is when and where the reaction is going to be happening. And when the reaction's complete, you're going to produce the products, and the products are going to then leave the enzyme. The substrate has changed, but the enzyme doesn't change at all. It can be used over and over and over again. So it's just going to be used, speeds up the reaction, and then the products can be made. A little bit more about proteins. The structure of the protein, the structure of the enzymes, because all enzymes are made of proteins. Not all catalysts are enzymes, only some catalysts are enzymes, but all enzymes are catalysts, and enzymes are made of protein. We learned, when we learned about biomolecules, that proteins have a chain of amino acids. The monomer for proteins is amino acids, and their primary structure is just a chain of amino acids. Well, proteins are very special because they do everything. And based on the chemistry of these amino acids, which you don't need to know until AP Bio or college level biology, biochemistry, based on the chemistry of the amino acids, the protein is going to fold up in a certain way. There's different ways the protein can fold. They can make something called, like, I try to draw it like beta sheets. They can make alpha helixes. And based on this structure of hundreds of amino acids long, there's going to be a 3D structure of the protein. The amino acids, I tried to draw a bunch of amino acids on there, are going to fold up onto each other and make a particular shape. And that shape is what determines how the protein works. And that shape is also going to determine how the enzyme enzymes that are, or proteins that are enzymes, how it's going to function. So this is its 3D structure. And when you move on to AP Bio or College Bio, you can call it the quaternary structure. What happens with enzymes is sometimes they can be denatured. When an enzyme's denatured, that means this beautiful 3D structure of a protein is going to unfold. It's going to unfold, and thus it's not going to be able to function like it was able to function before. Its shape has changed. Its active site is not going to work. It's not going to fit together with the substrate like a lock and key. It's just not going to be functional. So a denatured enzyme can happen from a couple of ways. The main way is a very high temperature. After we get to the best temperature for our enzymes to work, about 37 degrees Celsius, 98 degrees Fahrenheit, after a while, it's too hot and it's going to break this structure up, and the 3D structure is going to unfold. So it's going to unfold and it's not going to be functional. A very extreme pH, a pH that's too high or too low, could also cause the protein, the enzyme, to unfold, and thus it's denatured and it's not going to work anymore. Some other things that come up with enzymes is you could have an inhibitor. We saw in our activity that and with the online simulation that an inhibitor is physically going to be on the active site in the enzyme, and it's going to block the substrate. It's going to prevent the substrate of going into the enzyme, and thus it's not going to allow that enzyme to speed up the reaction of that substrate. So over time, the inhibitors are going to cause the reaction rate to decline. You could also have a coenzyme. Coenzymes all work in different ways, so it was really difficult to make a basic drawing of this, but basically a coenzyme is just going to help the substrate attach to the enzyme. So they can act in different ways, but it's going to speed up how the substrate and the enzyme bind. So over time, it will increase the rate of the reaction until we get enzymes and temperature. So we're going to see that temperature affects the rate of the reaction for enzymes when the temperature increases. So if I'm reading this graph, as the temperature increases, the reaction rate of the enzyme is also going to increase. And it increases until we get to this optimum temperature. And then for most, for our enzymes in our body and most living things, that optimum temperature is going to be 37 degrees Celsius, which is approximately body temperature, 98 degrees Fahrenheit. After we hit this optimum temperature, the reaction rate drops significantly. And this is because the enzyme is being denatured, it's unfolded, and it's not going to work the way it's supposed to. So the rate of the reaction rate decreases, and it actually should go all the way down to zero, because eventually all the enzymes will be unfolded, and it'll be too hot for the reaction to progress. pH is also going to affect the rate of the reaction for the enzyme. There's going to be an ideal or optimum pH for most enzymes. Most enzymes are going to have an optimum pH of 7, which is a neutral pH. Our blood is approximately a pH of 7. So you'll see that the reaction rate is really low, could be because of denatured enzymes. Before pH of 7, then it increases to our optimum pH, and then the reaction rate decreases again. pHs below 7 are considered acidic pH, and pHs above 7 are considered alkaline, or you can say basic, but the neutral pH is at 7. Some enzymes have a different optimum pH. For example, there's an enzyme in our stomach called pepsin that is going to have an optimum pH of 2, which makes sense because our stomach has a pH of 2. So the enzyme that's in our stomach, pepsin, is going to work best at that pH. But most enzymes, their pH is going to work best at a pH of 7.